with their anticodons. Let's see one of those guys. There we go. These transfer RNAs are made out of RNA, hence transfer RNA. And so I put a couple of RNA bases at the bottom to underscore that topic. These transfer RNAs float around in the cytoplasm and they hold amino acids. And one of the things they do is shuttle the amino acids to the ribosome. But there's something else, another important aspect of them. Their RNA nucleotides down at the bottom, at the very bottom of the transfer RNA, are always the opposite of the codon for the amino acid that they're holding. And what I mean by that is the amino acid for methionine is a UG. You think about, well, what's the complement to that? It would be UAC. There it is. And they call it the anticodon. So the bottommost part is always the anticodon for whatever amino acid they're holding. Maybe see if I can find another good example here. Any transfer RNA that's holding alanine, or I should say, if a transfer RNA has CGG as its anticodon, that transfer RNA will always be holding alanine, because CGG is the anticodon for the GCC alanine codon. Now it seems kind of strange at first, like why would these things have the anticodon for the amino acid that they're holding? Well, it's going to turn out that that's important for matching up the codon with that amino acid. There's the transfer RNAs holding the messenger, the amino acids. Here comes the messenger RNA. And here comes the first transfer RNA bringing the first amino acid in, and there's the next one. We're going to have a close-up now on what's going on in the ribosome. Here we go. All right, so notice that because the anticodon is complementary to the codon, they stick to each other. That's good old complementary base pairing. And so that ensures that the first amino acid that gets brought in is the one that goes with the AUG codon. It's kind of neat. That's a living thing system for making sure that the codons get read correctly. And the next codon, GAA, that's the codon for, what was it, glutamic acid. And so the transfer RNA that carries that glutamic acid has to have the anticodon CUU so that it will match up with that codon. Here it comes. And the next one's CCC. That's for the amino acid proline. So the transfer RNA that carries it in will have the anticodon GGG to make sure that proline gets matched up with that, that codon. And of course, there is no transfer RNA for the last codon because that's a stop codon. Yeah.
Are the transfer RNAs are amino acids?
No, they themselves are made out of RNA.
RNA?
Yes. They are made out of RNA. That's why they have RNA bases at the bottom. But nevertheless, they hold on to, each one holds on to one amino acid. Okay, so that's the basics of transcription and translation. What this lecture is going to focus in on mostly is the transcription part. Why the transcription part? Well, as it turns out, transcription is the key regulatory step in gene expression. If an organism decides it wants to express a particular gene or not express a particular gene, what that comes down to is, does it transcribe the messenger RNA or doesn't it, or does it not transcribe the messenger RNA? Transcription is this sort of the on-off switch, so to speak, for genes. All right, and the details of how the transcription works vary slightly between prokaryotes and eukaryotes. And so I thought I'd cover the prokaryotes first. They're a little bit more straightforward. Here's a prokaryotic cell. Remember, these the organisms that have prokaryotic cells are the bacteria, organisms within the bacteria domain, and organisms within the archaea domain. Archaea are a lot like bacteria, but they're classified in a separate domain of life. Anyway, what makes a prokaryotic cell is it doesn't have a nucleus. Its chromosome just sort of floats around freely inside the cytoplasm. Prokaryotic cells always have just one chromosome, and it's a circular chromosome, which is kind of interesting. I was kind of pondered, well, why is that versus the linear chromosomes of eukaryotes? I don't know exactly. Anyway, so they got these hoop-shaped chromosomes. So how many genes in a particular, in a prokaryotic cell? Well, it kind of varies from one prokaryotic cell to the next, but a typical bacteria might have around 3,000 genes. And so if we have 25,000 genes, what's that? About 1/8. They have about 1/8 the amount of genes that we do. Which is not bad considering we're big, complex creatures. They're just little simple bacteria. Okay, so let's say that this is one of the genes in the bacteria. And as I showed you on the board there earlier, whenever you have double-stranded nucleic acids, like double-stranded DNA, always the strands are anti-parallel, like you see there with the 5' and the 3'. All right, and let's just say that those 3 nucleotides right there are the first codon of this particular gene. And so this is going to be codon 2 right there, GCA, and then codon 3 is going to come next down the line there. And so in a sense you can say this gene has a polarity. The first codon's here, second codon's there, third codon's there, fourth codon's there, fifth codon's there. The gene runs in this direction to the right on this diagram. And so when I say that I would now want to look upstream of the gene, I mean in the direction away from the codons. So the region before the first codon, in this region right here. Upstream of the first codon in bacteria, you're always going to find a special nucleotide sequence called the promoter. And what's special about that sequence is that's where the RNA polymerase binds to. Because remember, the RNA polymerase has to bind onto the template strand so it can transcribe it into the messenger RNA. Well, how does the RNA polymerase enzyme find the beginning of the gene? It's this promoter sequence. The RNA polymerase has a binding site that looks for a particular sequence of DNA nucleotides, and it locks onto that particular sequence. In other words, the promoter is a sequence that says codon number 1 is very near here. And in bacteria, that's what the promoter sequence is. You don't have to memorize this particular sequence, but that's the sequence that the RNA polymerase looks for and binds onto. All right, so bacteria is going to express this gene, so the 2 DNA strands move a little bit apart. Here comes RNA polymerase. It finds that particular sequence, links onto it. It's got a binding site for that sequence. It links onto it, and now it has attached itself to the template strand, and it's just going to chug along it now, making the complementary mRNA. Chug it, 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 chug it. Take special note, as always, nucleic acids are always made starting at the 5' end and working towards their 3' end. Matter of fact, I think I stuck that verbiage in here somewhere under template strand. RNA polymerase enzyme the mRNA is always made from the mRNA's 5' end to the 3' end, which, because of that anti-parallel business I just mentioned, if you think about it, that means the RNA polymerase has to move along the template strand from the 3' end to the 5' end of the template strand. In other words, it's going to move along this direction in the template strand. So, another thing to note, there we go, the promoter is not right smack dab next to codon number 1. The promoter is actually a few nucleotides upstream of codon number 1. There's codon number 1, the AUG right there. The promoter is actually up here. And so when transcription begins, it's not right at codon number 1. There's always going to be a few nucleotides in the messenger RNA before you get to the first codon. Now, if you've been following this, you might think, well, wait a minute, isn't that going to confuse the ribosome? Because when the ribosome gets this messenger RNA, how does it know that this is not the first codon? Just to give you a preview of what something that's going to come up a little bit later, it works like this. The ribosome scans along the beginning of the messenger RNA looking for the first AUG codon. For some reason, that's a special codon, and it always means start the protein here. In other words, AUG is always considered the first codon. And that encodes the amino acid methionine, so that means every protein begins with the amino acid methionine. Why amino acid methionine? I have no idea. It's just one of the facets of the genetic code. And so anyway, the ribosome is going to ignore all these because none of those are an AUG codon. But when it finds this first AUG, it says, aha, that must be codon number 1. And so now it's going to make methionine, and then codon number 2, GCA, whatever that is, alanine, to the next amino acid, et cetera, et cetera. And that little sequence right here, that's 5' to the AUG codon. In other words, a little bit upstream of the AUG codon, is called the 5' untranslated region because it's part of the messenger RNA, but it's never translated into amino acids. It's essentially skipped by the ribosome. All right, so here's a kind of a zoomed out view. There's the promoter in that yellow box right there. There's codon number 1, and here's the ribosome making the messenger RNA. Now, what I want to address next is what tells the RNA polymerase when to stop making that messenger RNA, because it has to finish somewhere. We got to end this messenger RNA somewhere. If not, what would happen? Well, I guess it would just keep chugging along forever, and you would get a messenger RNA as long as the chromosome, which, you know, you're only supposed to be transcribing 1 gene at a time, so you would get all 3,000 genes transcribed, which would be ridiculous for 1 messenger RNA. So how does it end? Well, first of all, the messenger RNA, the transcription, does not end at the stop codon, the TGA codon. The RNA polymerase is going to go past the TGA stop codon and keep transcribing a little bit past it. There we go. Now there's the UGA stop codon in the messenger RNA. And so here's some things that are beyond the stop codon. But a little bit past the stop codon, there's going to be a series of U in the messenger RNA, because in the template strand is A. And there's something about that particular sequence, all those U's in a row, that destabilizes the RNA polymerase, makes it derail itself from the template strand. And they call that sequence the terminator, a special nucleotide sequence in the messenger RNA that causes the RNA polymerase to detach itself from the template strand. And thus that ends the messenger RNA, that ends transcription. And again, take note that terminator sequence is not right after the stop codon, the UGA. There's always a few nucleotides after that. And that's called the 3' untranslated region because it's 3' to the last codon. It's downstream of the last codon, but it's never translated into any sort of amino acids. Sound all right? Okay, so everything I just got through telling you is the way transcription works in prokaryotic cells. In eukaryotic cells, it's similar in many ways, but there are some important differences. And maybe just to begin with, we should refresh ourselves that eukaryotic cells have a nucleus. That's what makes them eukaryotic cells. But another difference is the chromosomes in eukaryotic cells are linear chromosomes, not circular ones. But the same concept of anti-parallel strand orientation still applies. All right, so here is a gene in a eukaryotic cell. Same basic setup, meaning that you have a promoter, which is where the RNA polymerase is going to attach. Actually, maybe I should point this out here, that the enzyme that transcribes the genes is called RNA polymerase II, or RNA pol2 for short. And so you might wonder, well, is there an RNA polymerase I? And there is. In fact, there's also an RNA polymerase III. And those enzymes do transcribe genes, but not protein encoding genes. Those RNA polymerase enzymes